Start recording. Okay. Lovely. It's working. Okay. So we're going to go through 1.2 and 1.3. I'm not going through any of the enzyme stuff, which by the way has also changed a lot since last year. But anyway, yeah, so I'm only going through the main cell respiration and photosynthesis part. The structure, I'm going to go through it for those who joined just now. I'm going to do cell respiration SL, cell respiration HL, so I'll tell you when I transition into the HL part. And then take a break, and then photosynthesis SL, and then photosynthesis HL. That's how I'll go through all of it. If you guys have any questions, keep putting them in chat so I can take a look and answer them as I go along. And I will, of course, also take questions at the end. Okay, so let's get started. I will record it and I will put the recording in the chat after. I am recording it and I will put it. Okay. Cell respiration. Okay, the first thing we need to know regarding cell respiration and photosynthesis, I guess, also, is ATP. So, before I start this, what is the full form of ATP? Everyone put your answers in chat, and please spoiler your answers before you put the answers in the chat. Yeah, this should be simple. Everyone should know this by now. What is ATP? Okay, so add a double, what I just put on either side. So add both on either side of your answer. Two on each side, but okay, yeah, so full form and what it is, I'm okay with both. A lot of people are putting the full form in, which is fine as well, but yeah, so yes, most of you are correct. Sakura, it's adenosine, not adenine, but yeah, so it's adenosine triphosphate, which basically is, as someone else said, energy currency of the cell. So it basically is what's used as energy in cells. It's what's used to perform cellular functions. And there are some properties that make it ideal as to be that energy supply. So firstly, it's chemically stable at the general cellular cytoplasm pH levels, which means that it won't break down and prematurely release energy and only releases it when necessary. Because if it broke down prematurely, then you just have excess energy that goes to waste, firstly, and then secondly, it wouldn't actually be useful for specific functions. Secondly, it's soluble in water. As we know, cytoplasm is mostly water, so it's really important that it is soluble in water so that it can diffuse freely in the cytoplasm, so that it can be used, it can move within a fraction of a second, and that means that it can be used immediately, essentially. pH changes doesn't cause it to break down. I am unsure, but also you don't really need to know it, but generally speaking, any protein would break down at certain, protein denaturation, right? So it's basically that's how it would work. So I mean, in terms of ATP, you don't need to know it, but you need to know that because it's similar to a protein, the fact that it doesn't break down at cellular pH, that means that that's like around the optimum pH, I guess. It doesn't have issues, if that makes sense. I'm trying to phrase this as best as I can. But basically, it means that at the pH that our body is at, or at the cells in our body are at, ATP stays chemically stable, and that just makes it that just makes it possible for ATP to be very specifically used, if that makes sense. I waffled a lot there, but basically that. What was I? Yeah, okay. So yeah, I talked about solubility in water. So the next thing is it is unable to diffuse through the phospholipid bilayers of our body's membrane. So whether that be the cell membrane or otherwise, everyone should know membrane transport, and we will go through it in another session. But basically, because it's unable to just diffuse through, our bodies and our cells can control how it moves between cells, and then there'll be no like unnecessary leakage of ATP outside of our cells. Next, releases energy sufficient for cell tasks. So when ATP breaks down into ADP, when you remove the third phosphate group, it releases like just about sufficient energy for cell tasks, with a little bit extra, which is wasted by transformation to heat, but it makes just enough where it can be used for cell tasks, even after the transformation energy is used, if that makes sense. Okay, next is easily regenerated. Again, ATP to ADP, which is something we will discuss in a little bit, is a really common reversible reaction, which means that it's really easily regenerated in order to make ATP again, and then break it down again, and then make it ATP again. And yes, I will put the slides up as well with the recording. Okay, now this is the other SL thing: aerobic versus anaerobic respiration. This can very easily be like I would say a 4-mark, 5-mark question, or I mean, realistically, I would put it at a 4-mark question, but it depends. IB's unpredictable sometimes. But it could just be like what are the differences between aerobic and anaerobic respiration question. So it would just, they would just expect you to write down the differences. So obviously, aerobic, anaerobic, the main difference is the presence of oxygen. In aerobic, there is oxygen. In anaerobic, there isn't oxygen. If you didn't know that already, what are you doing? You should be worried. If you did know that already, you should be worried. Okay, the next is substrates. So for aerobic respiration, the substrate can either be sugars, or it can also be lipids or triglycerides. It's not restricted to only carbs. If it's anaerobic, it can only be carbohydrates, so it can only be glucose and other sugars. Next is ATP production. So aerobic makes anywhere from 30 to 32. I think 32 is like kind of the standard number people always say, but like anywhere from 30 to 32 ATP molecules, and anaerobic respiration makes 2, or somewhere around like approximately 2. Basically, the difference is aerobic will make a really large yield, while anaerobic will make a small yield. Next is waste products. Now, aerobic, because oxygen is there, it forms, as its waste product, it makes carbon dioxide and water. For anaerobic, since there is little to no oxygen, it forms something called lactate or lactic acid. And no water, obviously. Now, so aerobic respiration happens both in the cytoplasm and the mitochondria of cells, while anaerobic will only happen in the cytoplasm. Okay, is that a good enough summary of aerobic versus anaerobic? Perfect. Okay. In the matters of textbooks, I would agree that the Oxford main textbook is a lot of waffle. But in like, is it 36 ATP? I don't know. Okay, this I picked up from the Oxford study guide and it said 30 to 32, but then some people say like 38 ATP. Like I was taught 38 ATP. But yeah, there's a like, it's not standardized. But yeah, is it that aerobic respiration only occurs by digesting sugars and lipids? I don't know what you mean by that question. Okay, yeah, so I'm gonna believe T, because yeah, so 32 to 38. Basically, the point is that it's a large yield. I'll change the 30 to 32 to 32 to 38. But yeah, the main point is that it is basically a matter of it being a large yield. Okay, about the substrates, basically, in order to break down lipids, it has to have like in order to have aerobic in order to break down lipids to use as energy, it has to be aerobic respiration. It cannot be anaerobic. For carbohydrates, it can be either anaerobic or aerobic, if that makes sense. Basically, it's a matter of carbs, sugars, glucose, etc., can be either aerobic or anaerobic, but lipids can only be aerobic respiration. Yeah, again. Okay, so now we're gonna do a little bit of interactive thing. Can people put in chat the word equation for aerobic respiration? Word equation, because apparently you do not require to know the chemical equation, which is blasphemy, but you know, what am I supposed to say? I'm not the one making the rules, so whatever. It was, I don't know, man. I'm going based off of the Oxford study guide, and the Oxford study guide just said the word equation, and I died a little bit inside when that happened, but you know, again. Which reminds me, yeah, textbook things that I was talking about. Oxford main textbook is definitely a lot of waffle. Like, it's just so much extra information that you really don't need to know. Cognitive is really good, but again, I've seen sections where there can be a little bit too much information as well, but that can be a personal thing also. But my favorite, when I was doing IB anyway, my favorite was the Oxford study guide, which is basically, it's made by Oxford anyway, but it's it's like really, really condensed. Like, it's just the information that you need, rather than it being like just a bunch of random information, if that makes sense. Nice. Everyone's pretty accurate. In the textbook, it does not say plus ATP, it just says plus energy. But I won't begrudge you of the answer if you say plus ATP, really. But yeah, so that's what it was in the Oxford textbook, I think. Okay, now the same thing but for anaerobic. But yeah, thank you. This is all like I basically, this one I've reused mostly from last year's slides, but I had to remove so much information. No, if you learn that it's 30 to 32, that is not wrong. It's different in different I'm going to put 30 to 38 now. It's different in different textbooks, and different resources say different things, but the main point is that whether you write 30 to 32 or whether you write 36 to 38, whatever, the main point of me putting that there is that it's a larger yield than anaerobic. So if you're in a test and you're like if you write 30 to 32, you're gonna get it right anyway. But if you're worried about that kind of thing, then you can always just be like, it's like 30, sorry, it's a larger yield. Yeah, sorry, I lost my train of thought there. But yeah, okay, we did Arabic already. We're doing anaerobic. Okay, have people put anaerobic answers in the chat. Okay, Sakura, the one you're writing at the top is for plants, which is, or sorry, yeast, I guess, which is not wrong, but also, yeah, sorry, I was focusing more on humans anyway. It's fine. It's not, you're not wrong at all, that's right, but, yeah, I realized I didn't specify whether or not I was focusing on, yeah. Okay, yeah, most of you have gotten it right. It is glucose to lactate or lactic acid, plus energy. You can write either lactate or lactic acid. I don't think they really care, but I mean, again, this is from the textbook, but it's still right. Yeah, so the glucose plus ethanol plus carbon dioxide plus energy is anaerobic, but in plants / yeast, anaerobic respiration. It's not for humans. That's the difference. So the lactic acid is made in us humans, and the ethanol and carbon dioxide thing is in yeast and plants. Yeah. Okay. Are we good? Let me see people's answers. I think everyone did get it right. I don't know if I saw one that was missed, but yeah, I think that should be good. Are we good with Arabic and anaerobic? I'll move on quickly if it is all good. Someone's typing, so I'm gonna wait. If there's questions. Aerobic respiration, can it break down sugars and glucose, and then anaerobic is sugars and only lipids? It's the other way around. So aerobic can, okay, I think the way I've phrased this is really wrong. Does that make it clearer? So for aerobic, it can be sugars and lipids. For anaerobic, it's only sugars. Humans, anaerobic can only use carbohydrates as the substrates and not anything else. Yes, it's just carbs. if you're asked to write the equation for anaerobic in the exam, would they take off points if I put ethanol instead of lactic acid? They'll usually specify. So they'll usually be like, it's not a dumb question, by the way, but if they would usually specify, they'd be like, okay, what is the... It also depends on context. So sometimes they'll do these like really long, like 6-part answers, and one of the parts, and it'll be about, for example, alcohol production or something, and the question will be, what's the equation for the anaerobic respiration in this situation. So when it's alcohol production, you have to know that it's ethanol and it's not lactic acid. It's more of a concept thing. So they'll I mean, in my experience, they'll only ever ask it in those type of situations where it's like a really long, like, for example, they're talking about alcohol production for some reason, and then there's like 6 different parts to the question, and then one of the parts is a 1-mark question, is what is the equation in of the aerobic respiration in this or they won't even say anaerobic, they'll just say what is the equation of the respiration shown in this situation. And then because it's alcohol production and they're probably talking about yeast, then you have to kind of infer that it's about ethanol and not about lactic acid, if that makes sense. Okay, next, this is basically where cell respiration for SL ends, I believe, unless I'm missing something. It's phosphorylation. So phosphorylation is the addition of a phosphate group to an organic molecule. Now, whether or not you need to know the fact that it's a phosphate group, I'm hesitant to say yes or no, because I've forgotten whether or not HL, SL differences. But basically, the point is that when you add a phosphate group to an organic molecule, it makes it less stable and therefore more likely to react in the next stage of a metabolic pathway, which is just a fancy way of saying next step in cell respiration or photosynthesis or otherwise. So it'll basically become, for anyone who does take chemistry as well, it'll basically go from a slow endothermic reaction to a rapid exothermic reaction. And the phosphate group is usually transferred from ATP. So it goes from, when you add glucose and ATP, you get glucose 6-phosphate. When you break down glucose with ATP, you get glucose 6-phosphate. So you're adding a phosphate, and ADP, which is where one of the phosphate comes off. So ATP becomes ADP because ATP is adenosine triphosphate and ADP is adenosine diphosphate, which basically means that it gets rid of the one phosphate from ADP to add it to the glucose to make it less stable so that it can do... This is, okay, this might be HL as well. I don't remember whether or not this is an SL. Let me double check, because I could be really wrong. I just remember having to do this last year, so I just repeated it. Let me just triple check. HL, SL, HL. Okay, you don't need to know this entire in-depth, but what you need to know as SL in terms of phosphorylation, you do need to know a little bit, but all you need to know is that ATP and becoming ADP is a hydrolysis reaction. ATP into ADP is a hydrolysis reaction, and ADP to ATP is a condensation reaction. That's what you need to know. I will modify this slide later on, but basically, let me just add this little screenshot into it. Where is my slideshow? Okay. You can ignore everything else about this section. It's just, you just need to know about this one. I will remove everything else, but you just need to know that, basically. So what you need to know is when you go from ATP to ADP, it's a hydrolysis reaction because you need the water to help split the phosphate bond, and then it becomes ADP. And then phosphorylation is when you add the phosphate back, so it becomes from ADP to ATP, with a little bit of energy, and then it becomes a condensation reaction because it releases water. Does that make sense? Sorry, I forgot the fact that it's not included in SL. Can I explain how the phosphate moves? Sure. Basically, ATP has 3 phosphates joined basically to adenosine, right? So it's called adenosine triphosphate, which I'm putting in chat right now. So that basically means that there's 3 phosphates in the most simplest of terms. I'm not saying this is extremely chemically accurate, but there's 3 phosphates joined to adenosine, right? What happens is when I'm trying to re-find my train of thoughts. When the water molecule splits, it also allows, you don't really need to know this in depth, but essentially what happens is that the ATP becomes ADP because one of the phosphates splits when water splits, and then one of the phosphate gets becomes a singular radical, and ADP is formed because there's only 2 phosphates now, and I've forgotten what the third bond becomes, but basically that happens. And then when you rejoin ADP and the phosphate molecule, you release water, if that makes sense. I don't know if that makes sense, but that makes sense. The Oxford study guide is enough to study off of, would there be anything I would be missing? When I did it, I did a mix of the Oxford study guide and Cognity, just because it just in case there was something that was missing. Where does the water come from? How and when does ATP react with water and then become ADP? So phosphorylation happens in, I'm trying to think now. It happens in, I don't think you need to know it as SL, but it does happen in, wait, I'm going to ask T. Is she here? She's here. Yes, she's here. T, help. ATP becomes ADP in the matrix. I don't want to get this wrong. Let me double check and I will get back to you. ATP to ADP. Yes, but where does it happen? It happens both in cytoplasm and mitochondria at different steps of the water comes from the fact that it happens in the cytoplasm. The cytoplasm has water, the mitochondrial matrix has water. Water just is there, if that makes sense. AMP. What's AMP? I don't know what you... Oh, no, then you don't. The answer is no. The answer is no. You don't need to know AMP. So when the cell wants energy, it'll complete this reaction with the water around it to create energy. Yes, is how I'm going to say that. See, all of that might be true, but as a SL student, you don't need to know all of that. It's all true, but you don't need to know all of it. You go as far as ATP is hydrolyzed, ADP and phosphate are produced. That's basically all you need to know. I never use Save My Exams. So I would, yeah. My favorite, where I took all my notes from, was the Oxford Study Guide. But yeah, you don't need to know the entire AMP thing in SL, as far as I know. BioLetters works. Again, okay, I feel like I've said this so many times, but my favorite resource was the Oxford Study Guide. As much as the Oxford textbook is like waffle, the study guide is very like comprehensive. I don't know about this year's. Last year's I'm talking about. I don't know how good this year's is. I can't vouch. But yeah. Okay. Let me move on really quickly. Okay, now everything past this is essentially just HL content. Let me double check if I've missed anything in terms of SL. I mean, other than the fact that you should know the differences between cell respiration and ventilation. I hope all of you know that already. I hope all of you know that respiration is what releases energy. What else? Yeah, I think that's it. Meantime, the HL section. Oh, variables affecting cell respiration. Oh, I forgot about that. I did not add it into my slides. But let's see. Okay, I'm going to ask the SL kids to answer this. What are the variables that affect cell respiration? Temperature, yes. B, you don't need to know about where the phosphate goes. If I'm being honest, they'll never ask you anything like that. All you really need to know is that ATP becomes ADP through a hydrolysis reaction, and ADP can become ATP with a phosphate molecule through a condensation reaction. That's essentially all you need to know. Temp, yes. pH, yes. Oxygen concentration, also yes. Substrate concentration, also yes. I think those are the main four. Let me double check if there's anything else. Oxygen concentration, substrate concentration, temperature, pH. That's about it, really, in terms of SL. There's enzyme concentration as well, but I don't think it's like, yeah, that's it. So yeah, that's right. That's all right. Okay, good. Is there anything else in SL that I've missed? Respirometer. Do you guys need to know how to use a respirometer? I swear they've never ever asked a question about the respirometer. It's not in the Oxford study guide, but I guess you should know how it works. Yeah, the experiment questions, that's true. They might just add it in and be like, this person used a respirometer. Okay. Let's see. A respirometer. Let me find a quick picture. What's a good, where is see all of this, like, who's still, because I don't remember it being in exams, but better safe than sorry, I would say, I guess. Let me find a good example. Copy image. Oh, okay. Well, the guide, there's no mention of it, but if teachers have brought it up, then I mean, if teachers have brought it up, you should know. But basically the point is that it measures respiration, rate of respiration in living organisms, and they quantify oxygen consumption / carbon dioxide production. And it just measures how fast it happens. Is that good enough? That's a very quick summary of what a respirometer. I don't know how to say it. You know what I'm talking about. But yeah. Good? Are we good? Yes? No? Perchance. Lovely. Okay. Now the rest of this is gonna be HL cell respiration. Okay, so HL has a lot more of the heavy lifting to do in terms of what you need to know for respiration. But first thing, oxidation and reduction reactions. All of you should know what oxidation and reduction is. Yes? No? Maybe so? Potentially? Roughly? Fine. Basically, in respiration, the oxidation of substrates means that you're removing pairs of hydrogen atoms, and reduction of a hydrogen carrier means that you're accepting the hydrogen from the substrate. So basically, yeah, if the SL cell respiration is done when I start SL photosynthesis, I will ping like 5 minutes before I start photosynthesis. SL. Okay. What was I talking about? Yes. So basically, substrates are oxidized, and then when it's oxidized, the hydrogen atoms are accepted by the hydrogen carrier, which for cell respiration is NAD, also known as nicotinamide adenine dinucleotide, which you don't need to know the full form of, I don't think. I just put it there, fun fact, if you're curious. But you don't need to write that in the exam. NAD is all you need to know. I was a very curious kid, so I googled what it was and I put it in my slides when I was teaching last year, and that's why it's there. But you don't need to know it. But basically, NAD is the most commonly used hydrogen carrier in our body, and especially in cell respiration. And essentially, it just accepts the hydrogen atoms when the substrates are oxidized. Yes? Good? Okay. Now we go into the more annoying parts. Okay, well, everyone should have a definition of cell respiration written down somewhere. But essentially, yes, you do have to know these reactions as part of HL. I believe so. I fear. Yeah, so cell respiration is the controlled release of energy from organic compounds, in this case glucose, to produce ATP. Cell respiration involves the production of ATP using energy released by oxidation of glucose, fat, or other substrates. If glucose is the substrate, the first stage of cell respiration is a metabolic pathway called glycolysis. Glycolysis is the first step when you're talking about glucose being the substrate in a cell respiration reaction. The same is not when it's another substrate, any other fat, etc. But we only worry about if glucose was the substrate. Okay, glycolysis, of course, there's enzymes in the cytoplasm, which, well, I'm glad, because you technically should know it if you're doing biology, because you should know basic cell respiration anyway. Even though all the details, okay, well, that's rude. She shouldn't have tested you on it, but you should know it. Like, it's not a bad thing to know, especially if you're doing bio, but yeah, testing you on it is a little bit like unnecessary, I would say. That was an old question, but I think, yeah, HL is more, it's more geared to HL students. Anyway, where was I? Yes, so in glycolysis, basically glucose is partially oxidized, and a small amount of ATP is produced here. Something you need to know about cell respiration is there is certain steps where ATP is produced even before we get to the actual Krebs cycle part of it all. So this is where a small amount of ATP is produced. The partial oxidation is achieved without the use of oxygen, though. So glycolysis is a step in both aerobic and anaerobic respiration. It's not a solo, it's not only for aerobic respiration, is what I mean. So basically, the most like overview, glycolysis basically converts glucose into something called pyruvate, which in anaerobic, when there's anaerobic respiration, the pyruvate is what's converted into lactate. But when it's aerobic, the pyruvate gets used in the link reaction and so on and so forth. Okay, so now we talk about all the nitty-gritty details of what the glycolysis reaction looks like. So basically we start with a hexose molecule, or glucose, and the first step is phosphorylation. So what happens is that 2 ATP get converted into 2 ADP, and then the 2 phosphate molecules from the ATP get added to the glucose, which forms hexose biphosphate, which, again, in the most simplest of terms, glucose and 2 phosphates. Like, it's just in the name. So essentially what this does is, like I talked about phosphorylation in the previous slide, it raises the energy level of the hexose, which makes it less chemically stable and therefore allows for the following reactions to happen. Without this step, it wouldn't be as like glucose is a very stable molecule. It would not be as easy to break down. The 2 phosphate molecules get added to the glucose. Are you HL or SLB? Sorry, I thought you were SL. Oh, you're HL. Okay, then you do need to know this. Basically, the 2 phosphates from the ATP get added to the glucose molecule. So glucose is also known as hexose, which basically means that it's like 6 sugars, 6 carbons, I think, is the point. But yeah, so because hex means 6. So the hexose and the two phosphates get added to the hexose, which makes hexose biphosphate, which looks like the same molecule, I know, but it basically just raises the energy level so it's easier to break down. Yes, decreases the stability, therefore makes it easier for it to be broken down in the next step. Which the next step is lysis or lysis, or I don't know how to pronounce it ever, but basically it just means splitting, which is why it's called glycolysis, or there's also electrolysis, etc., etc. Essentially just means the splitting of a molecule. So the hexose biphosphate, which is a 6-carbon compound, gets broken down into two trios, phosphate molecules, which basically means that there's 3 carbons and 3 carbons, and each of them have one phosphate group as well, which is what happens in lysis. After hexose biphosphate becomes triose, I don't know how to pronounce it, phosphate molecule, there's oxidation and then ATP formation that happens. So because of the splitting, there's 2 hydrogen molecules released, which get added to the 2 NAD molecules, which forms reduced NAD, or sometimes they'll write it as NADH+. But the point is that it reduces 2 NAD molecules. And then 4 ADP molecules, so as we know before, it went from 2 ATP to 2 ADP. Here it goes from 4 ADP to 4 ATP, which shows that 2 extra ATP were created here than were lost in the beginning, which means that the net energy released is 2 ATP in this glycolysis reaction. And the end result is pyruvate. Does that make sense? As a really, really quickly, I can re-explain it definitely. So essentially, all that happens, glucose turns into hexose biphosphate and 2 ADP are created, or 2 ATP are broken down, whichever way you want to write it. And then the hexose biphosphate gets split into 2 triose phosphate molecules, which then result in 2 pyruvate molecules as a result of the reduction of NAD and the formation of 4 ATP molecules. Does that make sense? Okay, so summary of glycolysis. 1 glucose converts into 2 pyruvates at the end of glycolysis. 2 ATP molecules are used per glucose, but 4 are produced at the end. So the 2 ATP that are used and then the 4 that are produced means that there's a net yield of 2 ATP molecules here. And then 2 NAD molecules are converted into 2 reduced NAD molecules. Is that good? Is that a good enough summary in terms of all the conversions that happen? So sometimes they can just ask you for example, what are the products of glycolysis? And then you have to be like, okay, 2 pyruvates, 4 ATP, 2 reduced NAD, and so on and so forth. Is that good? Are we good with glycolysis? Okay, I'm going to take no answer as yes. Oh, well, B answer. So yes. Okay, great. Now, before I go into link reactions and Krebs cycle and all the really, really, really difficult parts of cell respiration, I'm going to bring it back. The difference between anaerobic and aerobic respiration, I've been through this already, but one thing that you need to know in terms of glycolysis is that glycolysis occurs without oxygen, which means that it is the basis of all cell respiration, but also the basis of anaerobic cell respiration. So when glycolysis happens and pyruvate is created, that pyruvate either in the presence of oxygen will go through the normal aerobic cell respiration, or if there's no oxygen, the pyruvate can then become ethanol plus carbon dioxide in yeast or lactate in humans. So the pyruvate is was actually used to do the respiration part of all of it. The glycolysis is essentially just to make it easier for that process to happen, if that makes sense. So the pyruvate can only be oxidized further with the release of more energy from it if oxygen is available, which will occur in the mitochondria. So glycolysis basically occurs in the cytoplasm, and then the pyruvate is moved to the mitochondria, and then that's where the link reaction and the Krebs cycle, etc., 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 happen. But yeah. Okay. Link reaction. Okay. So, like I said, okay, give me a sec. Sorry. Yeah. So, like I said, pyruvate from the glycolysis in the cytoplasm is absorbed by the mitochondrion, singular. And then the link reaction happens here. So essentially, enzymes in the matrix of the mitochondria will remove hydrogen and carbon dioxide from the pyruvate. The hydrogen will be accepted by the NAD, and the carbon comes in from something called CoA or coenzyme A, which becomes carbon dioxide and forms acetyl coenzyme A. So basically what happens is pyruvate, this entire conversion is called oxidative oh my god, I always get this wrong. Oxidative carboxylation, yes. Since you're removing a hydrogen, which is oxidation, and you're removing carbon dioxide, which is called decarboxylation. So it's basically, yeah, you're removing hydrogen, you're removing carbon dioxide, therefore it's called oxidative decarboxylation. So after the link reaction, pyruvate and coenzyme A get together to form and release carbon dioxide, hydrogen, which gets accepted by the NAD, and then the coenzyme A and pyruvate without one carbon and one hydrogen forms the acetyl group, which then attaches itself to the coenzyme A, which then becomes acetyl CoA, which is then really important to use in the Krebs cycle. Is that a good enough summary of the link reaction, or did anyone not understand any part of it? Because I kind of butchered how I explained it, but yes. No, the enzyme is not the acetyl part. So CoA is the enzyme. It's coenzyme A. CoA is coenzyme A. And then acetyl is if you've done chemistry, it's basically a carbon group, if I'm saying an organic group, essentially. So the pyruvate has 3 carbons, but then loses one of its carbons and 2 of its oxygens to form carbon dioxide, loses 1 of the hydrogens to add to the NAD, and then therefore becomes an acetyl group. So just to quickly run through it again, pyruvate and coenzyme A. The point of coenzyme A is that it's always taken to the end. So the actual breakdown happens of the pyruvate. The pyruvate breaks down to the point where it loses carbon dioxide, which means 1 carbon and 2 oxygen molecules, and then also loses one hydrogen molecule, where the hydrogen then gets accepted by NAD, which forms NAD, reduced NAD, and the carbon dioxide is just released. So the pyruvate then becomes a 2-carbon molecule called acetyl, and then the acetyl or acetyl, I don't know how to pronounce it, gets attached to coenzyme A to form acetyl coenzyme A, or yeah, which is what we use in the Krebs cycle later on. Good, better. I don't know if that's a better answer, but are we good? Yes, Jess, are we good? Okay, perfect. Okay, so everyone's favorite part, Krebs cycle. So basically, this is where the acetyl groups are guys, I'm gonna mispronounce a lot of these words. Just like fill it in with whatever thing I say wrong. Acetyl groups from the link reaction are fed into the Krebs cycle. So basically, because it attaches itself to coenzyme A, the coenzyme A will deposit the acetyl group to a, will transfer the acetyl group, the coenzyme A will transfer the acetyl group to a 4-carbon compound called oxaloacetate. Oxaloacetate. Yes, what I said. Oxaloacetate. There we go. That's a 4-carbon compound, and then the oxaloacetate and the acetyl group will combine to form citrate, which is again a 6-carbon compound. Before I actually show you the diagram of the Krebs cycle, you need to know that there's 3 types of reactions that are involved throughout the Krebs cycle. There's decarboxylation, which basically means that you get rid of carbon dioxide, and this occurs in 2 reactions, which when this happens, it gets excreted together with the CO2 from the link reactions. There's oxidations, which is just, in this case, just the removal of just the removal of hydrogen. And then this will occur in 4 of the reactions within the Krebs cycle. And then it's accepted by NAD in 3 out of those 4, and FAD for the other 1. Essentially, FAD is just another hydrogen carrier, so it's just another version of the NAD. And these NAD and FAD both get reduced as a result. So when oxidations happen, the energy is released, which is then stored by the carriers NAD and FAD, which is later released in the ETC and chemiosmosis, which we will discuss after the Krebs cycle. And it's essentially, oxidation is what creates the energy to make ATP at the end. ADP production is another one of the reactions. ATP is only produced directly in 1 of the reactions, and then the rest of the ATP that's produced is produced in the ETC, or electron transport chain. And this reaction is called substrate level phosphorylation. Phosphorylation. I cannot speak. Yes. So this is what the Krebs cycle kind of looks like. It also has the addition of the link reaction right above it, but I've just put the whole thing there just so it, like, you can see how it relates to one another. So the top here is, I don't know if you can see my cursor or not. Let me see. Hold on. No, don't do that. Whatever. It's not gonna work. But oh, you can see me point. Lovely. So the pyruvate converting into acetyl coenzyme A is essentially just the link reaction. So as you can see, basically in these diagrams, the three circles or the three balls or whatever you want to call them are just the it's just pointing out the carbons. So what happens is the 3 carbons becomes its 2 carbons, which is then deposited to this, which is the oxaloacetate. Was it oxaloacetate? And then the 4 and the 2 together form the 6 carbon compound called citrate. But yeah, since it's a coenzyme A, it doesn't get used up in this reaction and therefore is always reused during cell respiration. But yeah, and then everything after this is the Krebs cycle. So what happens is the acetyl group gets added to the oxaloacetate to form the citrate. From the citrate, one breakdown happens where carbon dioxide is released, so 1 carbon's gone. And when the carbon dioxide is released, obviously there's also oxidation happening, which means there's hydrogen released, so NAD is reduced. That 6 becomes 5, obviously, since one of the CO2 have left, and then the 5 becomes 4 again, where another CO2, and it's basically a repeat of what just happened before this one. So carbon dioxide is released, hydrogen is released, since the 5 carbon compound is being oxidized, and then NAD gets reduced. Now we've gone from 6 to 5 to 4. Now from this 4 carbon compound, ADP and phosphate become ATP, FAD becomes reduced FAD, and NAD becomes reduced NAD. And these 3 reactions happen back to back. So this is where the number of reactions comes in. So there's 1 NAD here, 1 NAD here, and 1 NAD here. That's 3 reductions. Yes, 3 reductions or 3 oxidations, depending on how you look at it. It's 3 reductions of NAD, and then there's 1 reduction of FAD. There's 1 ATP production, and then there's what else? Decarboxylations here and there. Okay, do we need to know the name citrate and the other oxy name? I can't remember I remember it from when I did it, but I don't know if you need to remember it. So let me double check. If it says it on the thing, then you need to. Yes, you do. Oh, yeah. Oh, you can know 4C, 5C, 6C as well, possibly. It's in the Oxford Study Guide. So it says oxaloacetate and citrate. So I will write those down again. Oxaloacetate, citrate, and then, well, there's acetyl group. So basically, it goes oxaloacetate plus the acetyl group forms the citrate or citrate or whatever it's called. Some textbooks will also just say it goes from 4C to 6C to 5C to 4C to 6C again and again. But yeah, from in it says it in the Oxford Study Guide, so I'm just gonna run with it. How many reactions are there in total and how many of each reaction? I did say it in the previous thing as well, but basically there is 2 decarboxylations, so one here and one here. Okay, now whether you look at it as an oxidation or a reduction depends on what you're looking at. You can look at it, we mostly look at it through the point of view that the main glucose or the main compound or the main substrate is the reactions happening to that. So therefore we look at it in terms of oxidations rather than reductions of NAD or FAD. So there's 2 carboxylations, so 2 carbon dioxides are leaving the substrate. There's 4 total oxidations, 3 of which reduce the NAD and 1 of which reduces FAD, and there's 1 phosphorylation, or what's it called, substrate level phosphorylation, where the substrate basically allows for the condensation react, provides the energy for the ATP to be formed. Okay, so the first cycle is the link reaction and the second large one is Krebs cycle. Yes, basically. First isn't really a cycle. If you're talking about the first circle is part of the what's it called? If you're talking about the first circle, it's part of Krebs cycle. It's just from pyruvate to acetyl coenzyme A is the first. I mean, this circle is technically part of Krebs cycle as well. The only part that's the link reaction is from pyruvate to acetyl coenzyme A. After that, everything is still Krebs cycle. I don't know if I'm... Because the only link reaction part is pyruvate to acetyl coenzyme A. And then after that, the deposit of coenzyme A... the deposit of acetyl is technically part of the Krebs cycle, if that's... yeah. Which mostly they won't ask you specifically about Krebs cycle, I feel. I think if they end up asking you a question, it's going to be describe cell respiration. And then you have to go all the way from glycolysis to link reaction to Krebs cycle. So it's mostly interlinked anyway. Where I took this picture from also shows from glycolysis to the end. I just cropped it so that it's easier to understand. But yeah. No, you're fine, T. I think you're okay. Okay. Are we good with Krebs cycle stuff? If there's any... This image is from... Well, this specific one is from last year's... what's it called? Oxford Study Guide. But there's also a similar... There's similar ones everywhere else as well, I think. But yeah. I mean, it's a fairly common image. This one's from last year's Oxford Study Guide because, I mean, most of this presentation is from last year. I'm just carrying over. So, yeah. Yep, we're doing Krebs cycle. Are you able to link at the end of the session? I think the Oxford Study Guide is copyrighted material, so I don't know if I will be able to link the study guide, but I can. This is still AHL, this is not photosynthesis. I will ping people when photosynthesis starts, I promise. And then I think I can't link the study guide, but I can send this specific image and I will send it later. I mean, it's going to be on... It's like, I'm going to send the presentation anyway. But I can send this picture also if you need. I don't know what Solid means by biology flashbacks when he's literally an M26. Anyway. Okay, and anyway, what was I gonna say? Are you not doing bio this year? RIP bio. Anyway, what was I talking about? Yes, are we good with the Krebs cycle? Sorry, I keep getting distracted. It is HL. It is cell respiration. I will ping when I do photosynthesis SL. Sorry, I'm just getting distracted. Yeah, so we're good with this. The ETC. This pisses me off so much. I don't think you guys have this diagram, if I'm being honest, which pisses me off also, but yeah. Anyway, these were the pictures that we had in our previous Oxford study guide last year. And it's all just in one place. But I think in your study guides or in your textbooks in general, it's more broken down. I don't know. Yeah. Yes, you've seen it during the M24 study session. I did it last year. It's literally from the same study session. Is this diagram harder than the one we need to know now? I don't think so, in the sense that, well, I don't even know the diagram that you're meant to know now, but it's just basically, this is just all together. From what I can see in the study guide, they just break it down to the point where all you really see is like this section. But in M24 and before that, we needed to know the entire from the beginning to the end. Yeah, because T thinks it's simplified because T does biology in uni. R.I.P. R.I.P. IB biology. Yes, I will send it after the session. I mean, I'm gonna send this entire presentation after as well. I don't think you need the full diagram. You just need to know how it works. It's just, I prefer to explain with a diagram because it's a lot easier for me to go through it rather than go through every single reaction, if that makes sense. Can we ever be asked to describe the ETC? Definitely. Yes, 100%. They'll be like, what are all the reactions in ETC? I have definitely drawn this diagram before. It'll give you some plus points, I guess, in the examiner's eyes. Like, they'll be like, okay, yeah, they really know how it works, I guess. It just adds a little bit more. I prefer always to draw diagrams anyway in bio because visualization also helps how to explain certain things. So, yeah, that's just my personal opinion. Okay, enough procrastinating. How to do the ETC. Lovely. Okay, so where did we leave off? We left off with 3 reduced NAD molecules, 1 reduced FAD molecule, 1 ATP molecule here, etc., etc. But basically, we only really need to worry about the NAD and the FAD. So what happens is, these things are called, what are they called again? T-help, what are they called? I've forgotten. Membrane proteins. Never mind, I don't know why I'm asking T. Basically, you should all know this in terms of membranes and all of membrane transport and all of it. But essentially, this is the inside of a mitochondria. This is the mitochondrial membrane. This is the outside. It is the mitochondrial matrix, and this is the intermembrane space. And this entire thing is the mitochondrial membrane, but it's the internal mitochondrial membrane. So basically, that means, where's my picture? It's these ones, like what's it called? The little cristae, I think. It's essentially that. Okay. So. Yes. No, yes. 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 Why am I being dumb? Yes. Okay. So we start over here and we end over here. So it goes from 1 NAD, one reduced NAD molecule deposits hydrogen and an electron to the membrane protein here, and a reduced FAD molecule will deposit another electron over here, and then those two electrons together move throughout the membrane to essentially, what I like to say, cause chaos and basically create energy at this point. But yeah, so when that happens, when this reduction deposits the electron, there's hydrogen released outside in the matrix, right? Because the FAD technically had a hydrogen that it picked up from the Krebs cycle over here, and the NAD did as well. And then when it gets reduced and then when it gets oxidized again, it gets rid of its hydrogen, which gets released outside in the matrix, and it also gets rid of electrons, which then are moved through the membrane proteins, if that makes sense. And so when there's hydrogen over here, these are membrane proteins, which means that they have, like, certain molecules can transport through them. So hydrogen transports through each of these membrane proteins because there's a higher concentration of hydrogen through in the matrix because of these reductions, because of these oxidations happening. But anyway, so then once the electrons are deposited, they go from membrane protein to membrane protein essentially and move across the membrane until it reaches, the end of the membrane, where it the electrons are used to create water, where the excess hydrogen molecules in the matrix or oxygen and the oxygen in the matrix as well, both come together to form the water, and they accept the electrons from the chain. Now, after this has happened, while this is happening and the hydrogens are moving into the intermembrane space, since the intermembrane space is tiny, the concentration of hydrogen increases rapidly and then has to move, right? So because there's a higher concentration here and the concentration reduces out in the matrix outside, it moves through this protein called ATP synthase, which basically, synthesis, right? So it's synthase because it synthesizes ATP from ADP and phosphate. So these hydrogen molecules will basically go through this channel protein essentially and then generate enough energy to create ATP. Generate enough energy that ADP and the phosphate can be merged to form ATP. Does that make sense? Sorry, this is a lot. To what extent is it connected to the Calvin cycle? This one, not at all. This is in human cells. This is in the mitochondria. Well, it's in multiple cells, but this is in the mitochondria. Calvin cycle does not happen in the mitochondria. It happens in the thylakoids. Yes? No, maybe so. Anyway, and then the ATP is reused. Well, technically yes, because the ATP is used in all sorts of things. It can be reused in the beginning of the respiration, but it can also be used for other cell tasks. But because it's just another form of energy, is the point. A summary of the ETC, definitely. So, reduced NAD becomes NAD deposits an electron, also gets rid of hydrogen. Reduced FAD becomes FAD, gets rid of an electron, also gets rid of hydrogen into the matrix. These two electrons together from NAD and FAD move throughout the membrane through membrane proteins until it is accepted by the oxygen and the hydrogen outside in the matrix to form water. Since there's extra hydrogen from NAD and FAD in the matrix, how is it accepted outside? So oxygen is the terminal electron acceptor in the ETC in cell respiration. So essentially, if you know chemistry, in order for two hydrogens and an oxygen to form water, you need essentially to merge them. But because it's oxygen in the molecule form, in order to rip them apart, you need to deposit electrons. This is a lot of chemistry, but basically each oxygen has how many electrons? 8 electrons. But because they're it's unstable in a single atom, it exists diatomically, which means that it's always present as O2. But when it does that, it also loses 2 electrons, and it shares. Well, each of them lose an electron, and they share. They become a diatomic molecule, and then in order for the electrons to be accepted from the electron transport chain, they split apart and form normal oxygens again. This sounds like a lot of waffle. It's just chemistry. You just need to know that it's accepted by the oxygen. Some of what I just explained is probably also wrong, but that's also because I've forgotten a lot of my chemistry. Sorry. Yes, it's accepted in the matrix by the oxygen to form water. Yeah, and then the rest. So while the electrons are moving across the membrane proteins, there's an excess of hydrogen due to the NAD and FAD oxidations. So there's excess hydrogen in the matrix due to a concentration gradient. There's more hydrogen outside in the matrix. There's less hydrogen inside the intermembrane space. Is it? No, sorry, it's not that. It's basically you do... No. Okay, let me rephrase what I just said. Because the electrons are moving across the membrane proteins, there's enough energy to push the hydrogen molecules into the intermembrane space. And then, due to a concentration gradient between the intermembrane space and the matrix, because there's now excess hydrogen in the intermembrane space, because it's also a small space, which means the concentration is really high, the hydrogens get pushed through ATP synthase back out into the matrix, and when they're getting pushed out into the matrix, they essentially move the ATP synthase to generate energy, and that energy is what's used to form ATP from ADP and a phosphate molecule. In this case, it's not active transport. It's because of the move. T might be able to better answer this, but it's basically the movement generates energy, kinetic energy, which creates ATP synthase to move, which creates energy. Again, kinetic. It's all kinetic energy, which is then converted into chemical energy when ADP becomes ATP. Okay, does that all make sense to you? To all of you, I mean. Is that a good enough summary of the TC? Okay. In a very, very nice way, I've written down all the reactions that happen. So reduced NAD becomes NAD, which releases I was wrong, sorry. NAD will only release hydrogen. It doesn't release one of the electrons. FAD is what releases both the electrons. The reduction of NAD will only release energy, not electrons. Reduced NAD will become NAD when hydrogen leaves it, and also, obviously, as a result, energy will also leave. And then that energy allows for the movement of both hydrogen and the electrons. I think. Yes. No. Yes. Yes. I don't know. I'm like... Yeah. And then reduced FAD becomes FAD when hydrogen is released and 2 electrons are released, which cause proton pumping. So proton pumping is essentially where the hydrogens are being pumped into intermembrane space. So the energy from NAD pumps it here and here and here, but the energy from FAD will only cause proton pumping here and there, essentially, is what that means. And then again here, oxygen plus 2 electrons plus 2 hydrogens will form, half an oxygen plus 2 electrons plus 2 hydrogens will form 1 hydrogen molecule, but in a more simpler way, 1 oxygen molecule plus 4 electrons plus 4 hydrogens will form 2 H2O. And then ADP plus a phosphate molecule will form ATP. Chemiosmosis is basically the generation of ATP using energy released by the movement of hydrogen across a membrane, which is what happens here. The energy is used by the ATP synthase to form the ATP. Okay, now I think this is the last before we go. Oh no, okay, there's one more. And then we'll go into like a break, and then I'll do photosynthesis. So, there's some mitochondrial adaptations. Now, whether or not you guys still do this, let me double check. Because you might not have to do this anymore. Have you guys done this? I need a little bit help from M25 kids, because I don't remember. This was part of our syllabus. Well, yes, you'll definitely label the diagram. I mean, like, do you have to know about the adaptations of mitochondria that allow for cell respiration to happen? Like the different adaptations. Okay, so I'll still go through them then. Yeah, so there's multiple parts to a mitochondria. There's the outer mitochondrial membrane, which is basically like the actual like outer membrane of the mitochondria. Yeah, that's what I'm saying. This is not in the guide because this is from last year, but we had to do it last year. That's why I was like confused whether or not we have to do it this year. I mean, they have to do it this year. But according to them, they have learned about it, and I don't see a harm in really knowing, because it's like a few things only. But yeah, so again, some people might have done it, some, because I think also a lot of teachers are still reeling from last year's syllabus, so they might like just have done it just because they're so used to the previous syllabus. And some teachers didn't because they were sticking to the new syllabus. So there's probably a few inconsistencies there. I don't think it's too much to know, but if you don't really want to relearn this entire thing, you don't have to. It's really basic. Basically, the point is that there's some adaptations that allow for cell respiration in mitochondria to happen a lot better. It might be under another subunit as well. That might also be true. I could be wrong. Because this was under the same topic for us last year. But, where is it? Yeah, no worries. You're good. Let's see. Yeah, it did. That's the issue. Like, the topics got moved around a lot, and then they also got rid of a lot of things. So it's like, you know. But yeah, if it's not part of this subtopic, then I don't have to do it. Ooh, it's AHL B2.2.4. Well, okay, then I won't go through it. We'll go through it another time then, because it's not under the subtopic. But yeah, I guess you do have to know it eventually. Yeah, so this was the last thing I saw in the study guide, which I don't think expressly discussed last year, but I guess it is in your study guide this year, so I've added it in. The differences between when carbohydrates are used in cell respiration and when lipids or triglycerides. You do have to learn the adaptations. If you want me to go through them, I can, but it's just a different topic altogether. Yeah, I can go through them briefly. So basically, there's the outer mitochondrial membrane, which separates the contents of the mitochondria from the rest of the cell, and also creates compartments where there's ideal conditions for cell respiration, aerobic cell respiration, to happen, and then they create, what's it called, like the double membranes can be created from there. And then obviously the inner mitochondrial membrane actually creates the double membrane. Cristae, obviously, they increase the surface area, but also make really small, how do I put this? Really small intermembrane space, which allows for the concentration gradients to occur and therefore chemiosmosis to occur. And then obviously the matrix is really good in terms of it has all the enzymes for the Krebs cycles and the link reaction, etc. That's basically it. Okay. So, carbs versus lipids, like I was saying. So, one of the things is the ratio of carbon to hydrogen to oxygen and the impact it has. So essentially, in carbohydrates, it's a 1:1 ratio. That's a really funny sentence. So there's relatively more things that have to be oxidized, which means that it forms a slightly smaller yield of energy, while triglycerides have less oxygen, like way less oxygen. So there's relatively less to be oxidized, there's less oxidation, and therefore a higher yield of energy. energy that's released in carbohydrates, it's 16 kilojoules per gram of carbs, while for lipids it's 37 kilojoules per gram of lipids. The metabolic pathways used, so like I said, glycolysis is only the beginning. Glycolysis and a link reaction are only the beginning for carbs, to create those acetyl groups. But in lipids, when lipids are being broken down, there's something called beta oxidation. Now, you don't need to know how it works, you just need to know that there is a difference. So beta oxidation is what creates the acetyl groups for the rest of cell respiration when we're using lipids or triglycerides. And then there's type of respiration. Like I've said before, carbohydrates can have both aerobic or anaerobic respiration, but lipids or triglycerides can only have aerobic respiration. Is that good? Ratio part and how it's in relation to more or less ATP yield. So essentially, let me see how to phrase this. What's a good way to... I'm trying to think. Because there's... Let me... Hold on. I think you just need to know the fact that there's more in carbs than lipids. Basically, okay, so the way I will explain this is, in terms of ratio, there's more hydrogen in triglycerides than there is in carbs. Which means that, well, in terms of the compound itself, there's more hydrogen in triglycerides than in carbs, which means that it's less stable because it's it has more hydrogen, so there's less oxidation that has to happen, and therefore so less energy is used in that oxidation, and therefore more energy is actually released. Does that make sense? Like, in the sense that, okay, so in order for oxidation to happen, there's some energy required, right? But because there's less to be oxidized in lipids in relation to carbs, there's a larger yield in triglycerides than there is in carbs. You don't need to know more closely, you just need to know that beta oxidation is a thing that is different to glycolysis and link reaction, and it only occurs in lipids, because lipids don't go through glycolysis or link reaction. Yeah, no worries. Okay, are there any other questions regarding cell respiration? Yeah, carbs versus lipids was in the Oxford Study Guide, and I don't remember explicitly discussing it when I was doing it either, so I was like, let me just include it anyway. Okay, if there's no other questions, we're gonna take a break. If someone wants a challenge, you can try to draw alpha D glucose, but basically this is an opportunity for you guys to go stretch your legs, drink some water. I need water. I've been talking nonstop for the last hours or so. But yeah, I'm gonna take a really quick break. It'll be like 5 minutes. Yeah. Cool. Yeah, welcome back. I should do a ping as well. Let's see. I'm 25. Biology. Hi. I hope you guys still need to know how to draw alpha D glucose, because if you don't, I will cry. Yes, I can. Hi, welcome back. We're basically done with HL. We're taking like a 5-minute break so I can go get some water and stretch my legs as well. And then we'll get started with SL photosynthesis. But yeah. Is the one with H and OH where one is it the one with H and OH where one is either on top or below? That has to do with oh yeah, it is. That is. Yes. Sorry, I forgot what I was talking about. But yeah. You have to know the difference between, I hope you have to know the difference between alpha and beta D glucose, because the positions of the H and OH are changed. But yeah. Okay, I'm gonna stop streaming for a sec. I'll be back with some water and we'll get started in like, I want to say 4-ish minutes. Actually, I'm gonna give y'all a choice. Would you rather I do photosynthesis today or I do photosynthesis tomorrow? Because we do have poker nights in an hour, and I don't know if I'm gonna be able to do all of HL today. Well, I would start now, but tomorrow let me see. When's my other session? Around the same time as I started today. Yeah, around the same time I started today. Yeah, I'd probably do it, yeah, then, is my guess. Because I don't know if I'll be able to finish all of photosynthesis. I started at 2 today, so I'd spend 2 hours on cell respiration. English, yeah, I know, I'm doing English. I'll do that really quickly. But yeah. English is me as well. And bio is me as well. So I'll do bio from, I'll say 1 to 3 tomorrow. and then do English from 4 to whenever I end up doing it tomorrow. Again, all of these times are my times, but I will, hold on, let me, synthesis to tomorrow, I'm just doing so many sessions, guys. This is reminding me of all the biology I studied, which I haven't done in a really long time, by the way. Like, I haven't done bio in basically a year at this point. Because I'm currently a law student who has nothing to do with bio. I loved bio. Bio was my like my jam. I can't lie. Okay, let me also stop recording. I realize I'm still recording, but I'll put this recording